Today we are going to have the practical of the nematodes. For the diagnosis of the nematodes, first we need to consider the size, because we have the nematodes from a few millimeters up to more than 1 meter. The one which is more than 1 meter is Dracunculus medinensis, because it has been eradicated in most part of the world, so we are not going to see in the lab. The next, after Dracunculus medinensis, the largest one is Ascaris lumbricoides, which is the human intestinal nematode. When we talk about nematodes, because they are dioecious, so every single nematode is either male or female. So when you are going to look the adults, you need to consider if it is male or female. Ascaris lumbricoides, always the females, the nematodes, the females are longer. The females are longer than male. So the female of Ascaris lumbricoides is about 220 to 40 centimeters in length. The male, which is smaller, is about 15 to 30 centimeters in length. So here we are going to show you both male and female Ascaris lumbricoides. The largest one, which is also thicker than the other, this one is the female. The male is a bit shorter, and most important things about the male, at the end of the male there's a curve, and in this curve there's two nearly equal spicures that I'm going to show you in next slides. And this is the mouth of the Ascaris. They have 1 to 3 leaves, and this is the opening of mouth. Here you can see this is the female, larger and thicker, and this is the male. At the end of the male there's a curve. If we dissect the Ascaris lumbricoides, this is the female Ascaris lumbricoides, you can see the intestine, this is the intestine, and this is the reproductive system, the ovary. The female have actually 2 sets of sexual organs. In the female, nearly at 1/3 from the upper part of the body. You see, you need to consider the mouth from about 1/3 of the body. Here you can see the sexual belt. This is called sexual belt, okay? And in the ventral side of the sexual belt, there's a pore which is called genital pore. And from here, the eggs are shed in the intestine of human. So in the female, you need to find the genital belt nearly at 1/3 of the body from the mouth. And in the ventral side of the genital belt, you need to find genital pore. In the male, in addition of the curve, in the curve there is, as you see, 2 equal spicules. Actually, the spicules are the accessory organs of the male reproductive system. Okay, after fertilization, female through the genital pore that showed you release the eggs. When the eggs are released, they are not segmented. For the development of the larva, they need to spend sometimes in the environment in optimal conditions. This embryonated egg containing the L1 larva is the form which is found in the soil, but in fresh stool samples, the eggs that we can find is like this. This is the egg, which the size is important. Actually, like the adults, the size of the eggs, the size of the larvae is important for the diagnosis. The egg of Ascaris lumbricoides is about 45 to 75 micrometers in length. Inside the eggs, you can see the ovum, and it has different layers, inner layers, middle layers, outer layer. The outer layer of Ascaris lumbricoides is mammillated. It means it's not smooth. There's our projection like actually mammillated layer, which is characteristic for the egg of embryonated egg of Ascaris lumbricoides. In every egg, also in the eggs, you don't see any reproductive system, not so much characters, but usually there's at least 1 or 2 characteristics features which are very important to distinguish. Is it, for example, the egg of Ascaris lumbricoides or other nematodes or helminths. So for ascaris, this mammaliation is very important. Okay, sometimes the female is single and release the eggs. They are not embryonated. Or sometimes the females are actually releasing the eggs before copulation. Such eggs are called unfertilized eggs. It means they don't have any embryo. They cannot develop to the infective form. But it is important if we see such eggs in the stool of a patient, it means he is infected with Ascaris lumbricoides. But the eggs I'm going to show you today is the characteristic egg of Ascaris lumbricoides, which are fertilized. It means they can develop to infective form in suitable condition at the soil. Okay, when we talk about the size, we had Dracunculus medinensis, about 1 meter, Ascaris lumbricoides, about this size. After that, the nematodes, which is largest, is Trichuris trichura, which has a long, narrow part and a small, short part, like the whip. That's the reason it is called whipworm or soot. Okay, you say soot. Okay, Trichuris trichura has, as you see here, long, narrow part, a small, thick part. The one which has a straight end without curve is female. But in the male, at the end, you can see a curve, all right? In this curve, there is one single spicule with sheath. This is the female. As you see, at the end, it is straight, no curve. This is the end of the female. In both male and female, reproductive system are confined to the thick part of the nematode. These narrow parts actually contain the esophagus, a very thin esophagus. This is mouse, simple mouse esophagus, but for here, the thick part contains the reproductive system. Here, it is female reproductive system of the female, including ovary, uterus, but in the male, there is a curve at the end, very characteristic curve. And in this curve, you can find one spicule, one single spicule. So in the male of the nematodes, presence or absence of the spicule, the number of the spicule, the size of the spicule, the shape of the spicule is important. For Ascaris lumbricoides, we see two equal spicules. For Trichuris trichura male, there's just one single spicule, but covered with a sheath. That's the characteristic of male of Trichuris trichura. Okay, the female release the eggs. The normal color of the eggs like this are orange. When I say about the, for example, this is brown or orange, it's the natural color. Sometimes the samples may be stained in the lab and they have other color, for example, pink color. When I talk about the color, I mean the natural color of the egg. So it is orange in shape and they have two polar thickenings, two prominent thickenings at both sides. You see these two polar thickenings leads to a shape like barrel or lemon. So the shape of the eggs of Trichuris is barrel shaped or lemon shaped, or I can say it is like a tray. It is like a tray with two handles. Okay, so the egg of Trichuris trichura is like a tray with two handles. It is so characteristic because I've seen in the practical exam many times medical students ask me, okay, professor, will you set for our exam the egg of Trichuris trichura? Why? Because it is very easy to diagnose, and actually it's not going to be confused and mixed with other eggs. So it is very characteristic. You need to consider it is lemon shaped and it has two plugs like a tray with two handles. The size is important. The size is about 60 micrometers in length. So for Trichuris trichura, you need to see the male, the female. If you want to see the spicule of the male, you need to see under the microscope, and also the egg all the eggs, all the larvae can be seen by the microscope. We cannot see any egg without the aid of the microscope. Okay, for the hookworms, if we consider the size, okay, we have Ascaris lumbricoides, more than 20 centimeters, Trichuris, several centimeters. When we come to hookworms, they are about 1 centimeter and less. Ancylostoma duodenal, which is a bit smaller, has buccal capsule with 2 sets of teeth. Actually, 1, 2, 3, 4, 4 teeth. So when we say hookworms, they have buccal capsule. In the buccal capsule, we need to consider if there is teeth or actually cutting plates. In the necator, it is a buccal capsule with 2, crescent shape cutting blades. But here 2 sets of teeth. So by the marks you can see distinguish is it necator or ancylostoma. When I'm talking about mouse, about cuticule, it means the outer layer of the nematodes, or when I'm talking about the alimentary canal characteristics, they are nearly similar in male and female. When I say hookworms have buccal capsule, it is both for the male and female. The differences between male and female actually it is related to the reproductive system. Okay, this is the oral part. Near at the end, in both of them, in the male, they have copulatory bursa. Actually, copulatory bursa, like spicules, are supplementary sexual, actually reproductive system of the male. They are useful during the copulation. The copulatory bursa likes an umbrella. It's an umbrella which is open and they have rays like finger. Okay, it means they have several fingers. Okay, both of them has copulatory bursa, but in the necator, the spicules, actually the spicules are very long and narrow. As you see, these are 2 spicules and they are long and narrow, and the spicules in the necator usually are fused. It means they are combined. Today I'm not going to show you the copulatory bursa of the ancylostoma because it will be very difficult. You just need to see the opening or buccal capsule because it is easy to discriminate these two, and just to see the copulatory bursa of Necator americanus, which you need to compare with the copulatory bursa of the other nematodes, Trichoestrangulus species. In Trichoestrangulus species, spicules are not so delicate and narrow. Actually, they are small, short, and dark brown color. This is the mouth of ancylostoma. You can see the teeth. This is the copulatory bursa. These are the rays. Okay. Actually, end of the male is expanded, and in this expansion we see the finger-like projection. Overall, they are called copulatory bursa. This is the Necator americanus. You can see the cutting blades. End of the female, in the female, no copulatory bursa, and this is end of the male. The egg of both species of hookworms are morphologically identical. You just write hookworm egg. Nobody can say, is it the egg of Necator or ancylostoma? So morphologically, they are the same. All right. They have a shell, okay, which is smooth. As you see, it is smooth. There's no mammaliation. There isn't any polar plug. Smooth and thin. When these eggs are released in the environment, the larva is developed. The filariform or artery larva of hookworms is sheeted. Today, for the hookworms, I'm going to show you the egg, the mouth opening of the or buccal capsule of the ancylostoma duodenal, the buccal capsule of Necator americanus, and the end of the male in Necator americanus. The next nematode is Trichoestrangulus species. It means different species. The size is just a few millimeters in length. They don't have any buccal capsule, you see. The mass is very simple. This is the neck area. You can see the place of expiratory pore. And in the female, they have ovojector. It means part of the female reproductive system, which leads to push the eggs to shell in the intestine of the house. And this is the male. The male of Trichostrongylus species have copulatory bursa, like that of hookworms. But in the hookworms, the spicules are long, narrow, delicate, and most of the times they may be out of the nematode. But here, for the Trichostrongylus species, you can see two thick, dark brown, and short spicule. They are always in the nematode, you see, mostly in the nematodes. So in Trichostrongylus, I'm going to show you the end of male. You need to see the copulatory bursa. These are the rays, and these are the spicules, two long, short, dark brown in color. And in addition of the spicules, they have also gubernaculum, which is sometimes difficult to see. So consider actually the two dark, thick brown spicules, which are different from that of hookworms, because in the hookworms they are very delicate, long, narrow, and sometimes out of the nematode. The egg of Trichostrongylus is somehow similar to hookworms, and sometimes in the diagnostic laboratories they may be mistaken, but there is just a very small difference. In the egg of Trichostrongylus, they are a bit larger. They are a bit larger, as you see. They are about 80 or 90 micrometers in length, but not always. If when you look through the microscope, if you want to know the exact size of the eggs, the microscope that is using to needs to be calibrated. It means it should be set to measure the exact size. Is it, for example, 40 micrometers, 45? Okay. They are a bit longer, but the most important things, the egg of Trichostrongylus is asymmetric. It means one side is pointed, the other side not. It looks like the egg of hen. It is asymmetric. But the egg of hookworms are symmetric. It means it's not pointed in one side, and it is just a bit smaller. But today, because it is very difficult to discriminate, is it the egg of hookworm or Trichostrongylus? I just going to show you the egg of hookworms. Okay, don't worry. Right, the next nematode I want to talk about is Enterobius vermicularis or pinworm. The female are about 1 centimeter, the male is smaller, about 0.5 millimeters in length. Okay, I told you when I'm talking about mouth part or cuticule, esophagus, it is the same both for male and female. Okay, here at the mouth part, you can see cephalic alley. Okay, at the head of Enterobius vermicularis, there is cephalic alley. This is the esophagus, this is the intestine, but before the intestine, at the end of the esophagus, you can see a characteristic ball. You see, there is a characteristic ball in the end of the esophagus, both in the male and female. But after this, if you look through the, for example, here, this one, you can see too many eggs because it is female. So in the female, in the middle part, you can see many eggs in the uterus. And the other things for the female, it has a long, narrow part at the end, like a tail. That's the reason it is called actually pinworm, because it has a very long, pointed tail. So in the female Enterobius vermicularis, you need to consider the cephalic alley, the bulb in the esophagus, the eggs in the uterus, and the long, narrow, pointed tail. But the egg, excuse me, this is the male of Enterobius vermicularis. The end of the male is curved, and in this curve there's one single spicule. But it's not included in the samples I'm going to show you. Here, this is the egg of Enterobius vermicularis. Somehow we can see the eggs are like D, English D, the letter of D. The other actually helminths which have D-shaped egg is Dicrocelium dendriticum. In many cases, I found medical students actually misdiagnose these two samples, the egg of Dicrocelium with that of Enterobius vermiculans. First, you need to consider the color. The color of the egg in Dicrocelium dendriticum are dark brown because the adults are living in bile ducts, okay? So they have a dark brown color. But here, the eggs are released in perianal area, so they are colorless, they are transparent. If sometimes you see, for example, like today, if they are pink color because they have been stained, but the normal color is actually transparent. The other things, in one side, they are flat, other side there is a curve or convex. This convex is very similar to that of Dicrocelium. Inside the egg, you can see the larva, and it doesn't have any opercule. But the egg of Dicrocelium has opercule, so you can distinguish these two eggs from each other. The size is about 50 to 60 micrometers in length. For the Enterobius, today you are going to see the adult female under the microscope. Notice to the cephalic allen, notice the esophagus bulb, notice the eggs in the uterus, and notice the line pointed tail. And also you need to see the egg of Enterobius vermicularis. For Strongyloides stercoralis, there's no male parasitic male. Okay, so we don't have parasitic male. The female are embedded in the mucosa of intestine. What we see is the larvae in the stool. The diagnostic larva is L1 or rhabditoid larva, which is about 200 micrometers in length. When we talk about the eggs are smaller, okay? Smaller than larvae because it is just one cell. Many helminths has the eggs less than 100 micrometers. For some trematodes like Schistosoma fasciola, they are more than 100 micrometers. But when I'm going to talk about larva, for example, rhabditoid larva is about 200 micrometers in length. What about the internal organs? Larvae, it means juvenile stage. It means they don't have reproductive system. So just here, in case of nematodes, you can see the mouth, esophagus, intestine, and anus. At the end of the esophagus, in the L1 larva, there is the bulb in the esophagus. Okay, it's not clear, is it male or female, but the genetical material is embedded here. The genetical material is going to develop either to the male or female. Okay, here it is called genital primordium. It means the cells related to the reproductive system are here. If you see genital primordium nearly in the middle part of the L1 larva, it is related to Strongyloides stercoralis. Because the L1 larvae of hookworms, they have a very small GP which cannot be seen with ordinary optic microscope. So for Strongyloides stercoralis, the sample I'm going to show you is the L1 or rhabditoid larva of Strongyloides. You need to notice the GP in the middle part of the body. When the larva develop to infective form or L3, the size is nearly 3 times higher. It's about 600 micrometers. It doesn't have any bulb in the esophagus. The esophagus is large and straight, but at the end, it is notched. And I told you the L3 larvae of hookworms are sheeted, but here there isn't any sheet. So no sheet and no bulb because it's L3 or filariform. The characteristic feature of L3 or filariform larva of Strongyloides is the tail, which is actually notched. It means it's not pointed. Okay, the next nematodes are Toxocara. Toxocara, canis and cati are in the intestine of dog. But why it is important for us as a medical students, because the egg of Toxocara can develop to visceral larva migrants in human. Okay, so the adults of Toxocara, which are several centimeters in length, for example, up to 18 centimeters, are living in the intestine of dog and cat. Like other nematodes, most of the nematodes, the male is at the end of the male is a curve and there is a spicule, and the head of both of them have cephalic allele. The egg, actually these eggs are found in the stool of the dog. If you have pet dog or cat and you are worried if they are infected or no, so by a stool examination and finding the eggs, you can distinguish he or she is infected with Toxocara canis or cattle. Actually, the egg, the surface, the outer surface of the Toxocara egg is pitted or mahfur. You understand? Am I spelling correctly? Mahful. It means it is pitted. There's a lots of actually holes on the outer layer of Toxocara eggs. It looks like a golf ball or like some trays which have actually at the side like pitted. So the size is important. It's about 75 to 85 micrometer in length. It means less than 100. And the presence of such pitted shell is characteristic to the egg of Toxocara, which can be found in the egg of dog and cat, like hookworms, that we cannot say, is it the egg of Necator or Ancylostoma? Here, we just say the egg of Toxocara. We can't say, is it the egg of Toxocara canis or cattle? Just the egg of Toxocara. The last nematode we want to talk about is Trichinella spiralis, actually Trichinella spp. The male are just about 1 millimeter in length. Female is about 3 millimeter in length. Female actually is viviparous. It means they actually inside the uterus, the egg develop to the larvae, and from the genital cord, the larvae is shed. So it is viviparous, and the larvae migrate to the muscles. This is the end of the male. The male of Trichinella doesn't have any spicule. Instead of a spicule, they have two alae. But what I'm going to show you for the Trichinella today is the larvae in the muscles, okay? These are the striated muscles. Also, the larvae migrate to different parts of the body of infected host, but only in the striated muscles they develop the encysted larvae. Here you can see under the microscope the section of the striated muscles, including several larvae. Each larvae is cold and it is in a cell which is called nurse cell. The nurse cell is through the muscles which the larvae is embedded in. So for Trichinella, we just have the section of the muscles, including several cysts embedded in the muscles. And for the Toxocara, you need to see adult male and female like this, and also the egg under the microscope. If you have any question, ask me. Otherwise, I'm going to see you in the lab. Pardon?
Size, numbers. Is it important for fertilizer?
Size?
Yeah.
For practical, I just want you write me the name of the parasite.
And
I'm not going
size
to ask the details. No.
in the theoretical.
In the theoretical, I'm not going to ask the details. I may, for example, you should know the size. Is it at the millimeters, like Trichinella strongyloides? Is it at the size of 1 meter, like Dracunculus?
Okay.
But I'm not going.
But exact number?
No, I'm not going. For example, if the size of the male Trichinella is it 1.1, 1.6? No, just the approximate size. Okay, fine. You ask him, he asks your question. Okay.
This is the function of copulated spicules, and by tracking
Okay. Let's say I tell you the first one. In the lab, I can show you, for example, if it is the female, here there's a pore and genital actually birth. Genital birth is some part that showed that male need to grasp the female. By the aid of the curve, the male grabs the female, and SPQs help to open the vagina, and the S.U.M.S enter. They are very clever. The other,
The other one with the
trichinella. Trichinella. You see the presence of the spicules and, for example, governaculum, intricoestrangulus, it's not actually the real reproductive system necessary. For example, the female needs uterus, ovary. The ovary is necessary. In the males, the testis is necessary. The others are supplementary part. So in the trichinella, instead of actually spicules, they have a flap. The flap has the same role of the spicules. That's actually differences in the morphology. Yeah, accessory parts of the body. You're welcome.
Tissacara have many shapes. The egg of the Tissacara have many shapes. Have three shapes.
Have a what
Have
shape?
three shapes. Tissacara.
I know, but understand what you mean. Pitted, golf.
Have three shapes.
Oh, this is scan microscopy. Let's I show you. Excuse me. There's a question I just want to clear. You here you see three shape for Toxocara. This is the egg with larva. This egg is happening in the soil. When human children digest the food contaminated with the larvae, which has the larva, egg with larvae, not this one. The eggs which are passed in the stool, they don't have any larvae. For the development of the larvae, they need to spend some time in the soil. That's they are called soil transmitted hermits. So this is the egg with ovum. This is the egg with a larva, which is infectious for human. This is the egg in the scan electron microscopy view. Okay, so this is the egg with scan electron microscopy, but this one is the view you can see under the microscope. Okay, that's the difference. You're welcome. And here just this one. That's the one you are going to see is this one. The egg are found in the fresh stool. This is just to show you that the outer layer, which means pitted, it means like this. Or in this view, it is like this. You're welcome. Any question?
The task said that you said if anyone can draw slides and send them. The slides of the lab.
Who said no?
The task said that you said that whoever draws beautiful slides, that can send it. Is there specific slides, or we can't choose anything?
Whoever draw beautiful. No, I didn't mean. She couldn't understand what I mean.
Oh, okay.
During the whole course, I find one of the students, one of the girls, was drawing every picture very carefully with the details and everything. Anything we ask are, for example, MSc student cardiology or PhD students, she was doing lots of time to draw. I said for that special students, I'm going to have consider points because for spending so much time for this, not all the students. Okay,
So
because we are at the end of the course.
It's an offer.
I think she couldn't get my
Okay.
because her Persian is not, for example, as high quality as, for example, some of the other Taras. Okay, she couldn't get my message. Okay, thanks for asking, and then you clarify for others.
I will tell
Okay. Because I didn't ask her. I thought I saw every session he's going to draw and for herself, and she didn't ask me. I proposed. I proposed to Taro because she is going to spend too many time to draw the picture and coloring and drawing, and it is what we ask our PhD students, and sometimes they don't do. So I consider just points, positive points for being so careful. Otherwise, every students are spending the time. That's it. Okay. Okay. I'll see you in the lab. No question.